One of the common questions that comes up is what new research there is and what's going on in cutaneous lymphomas. And a lot of this pertains to lymphomas and cancer in general. The ability to look at the genes that are changed in a cancer cell have increased tremendously in recent years. And they've increased in terms of the speed with which we can look at a large number of genes, and also there's a significant reduction in cost. So where some years ago it would take a long time and be very expensive to look at individual genes, we can now look at many genes that are changed in a cancer cell relatively inexpensively. So that's starting to generate tremendous amount of information that may lead to the whys, which is often a very hard question, why people get lymphoma or what's going on in the lymphoma. So one of the areas that's going on now is we're looking broadly at a number of lymphomas, including cutaneous lymphomas, to see if there's frequent mutations or changes in the genes and try to get a better understanding of that disease. The hope is if there's mutations that we see recurrent in a certain disease, say like mycosis fungoides or another lymphoma, that you could then either have a therapy that targets that change, or that could lead to the development of a targeted therapy, something different than chemotherapy and more targeted to the underlying mechanism of why the disease develops. So I think that's probably the area that's going on most actively now and most likely to change therapy in the next couple of years.
There's a lot of research efforts, a lot of centers focus on genomic sequencing. And there are various types of molecular techniques that can be applied. And roughly there are so-called DNA microarrays. Those tests check for mutations. MicroRNAs, these tests check for RNAs that actually can silence certain genes. And then there's this group of comparative genomic hybridization that has been used to find chromosomal aberrations. So they were all very, very helpful. And they found a lot of mutations and a lot of aberrations. So now there has been a switch in so-called a whole genomic sequencing. And this sequences basically the whole DNA, the whole genomic information in such a tumor cell, which is a lot of work. And research is coming out. There are more and more centers who's doing that. It's extremely expensive. That's the downside of it. But hopefully these tests are sensitive enough to find clusters of mutations in certain subtypes that can really help to tailor medication, a treatment. So some mutations have already been found out, and treatments have been subsequently developed to address those mutations. Some other centers focus more on the T cell itself and how the T cell can switch and how the T cell is trafficking and circulating through the body, and why it's in the skin and sometimes in the blood, and why it's in both, and why some patients never have it in the blood and some patients just have it in the blood and not in the skin. So it's very complicated, and they focus on certain immune cell markers on the surface of those cells. Those immune markers can also be used for treatments. Some others focus on the tumor microenvironment, meaning other immune cells, and how these immune cells work and align with other immune cells to actually either suppress or make the tumor cells grow. Then there is a spectrum of translational research where other centers look into effects a treatment causes in the skin or in the blood, and see what's happening before and after the treatment that will help actually to find out and to learn more about the disease. So being in the field for now almost 14 years, so I know that has been done a lot of research and a lot of advances. When I started out, we basically just knew that mycosis fungoides was a CD4 positive T cell, and not many mutations have been found. But now the research is really helping us in guiding and learning more about subtypes and why certain subtypes can grow and why certain subtypes just stay and be very indolent.